Hello and welcome to another video lecture on my YouTube channel. Today in this lecture we are going to discuss about chromosomes. In this lecture we will discuss various structural elements present in the chromosome, and we will discuss how these structural elements like centromere or telomeres are essential so far as the function of the chromosome is concerned. This chromosome term was first introduced by Waldeyer in 1808, and this chromosome term literally means the color body. As in Greek we have this chroma which means the color, and this soma means the body, that is a body which is having a color. But here it is important to mention that the chromosome in itself is not having any color. We use special dyes in various staining procedures, and these dyes get chemically bind with this chromosome, and when we observe these chromosomes bind with these special dyes, they appear colored under the light microscope. So actually it is the color of the special dyes, not the color of the chromosome. So this chromosome means the color body. So let us first try to understand what are the various structural elements present in the chromosome and what are the various functions these structural elements perform. This is the general structure of the chromosome. The chromosome is having a telomere here, and then it is having a centromere, and then again it is having this telomere. That is, we are having two telomeres and one is the centromere. And here this chromosome structure is the structure of the chromosome present in the metaphase. So if you consider the cell cycle, we are having the division phase and then we are having the interphase. And the chromosome in this structural form is present only in the dividing cell. That is, a cell which is going under the division. Only in that cell you can find the chromosome like this. And in the non-dividing cell you cannot find the chromosome in this structural form. For example, this is the cell cycle. For example, let us consider the mitotic division. And here we are having this G1 phase. Then here we are having this S phase. And here it is G2 phase. So these are various stages. These are various phases of the cell which is undergoing the division. And here in G1 phase, S phase and G2 phase, the genetic material is in the form of the chromatin. That is, it is in the form of a thread-like structure. In this phase, the genetic material, that is chromatin, gets condensed. And it is the chromatin material in the most condensed state that it appears like this. That is, the chromosome is the most condensed state of the chromatin. And as you know, in the mitotic division phase, we are having four stages. That is, we are having here prophase, then we are having here the metaphase, then we are having here the anaphase and telophase. So these are four stages of this mitotic division phase. And if you consider the shape of the chromosome during this whole cell cycle, you will find that in these interphase or in the G1 phase, S phase or G2 phase, this genetic material is in the form of the chromatin, or you can find it just like a thread-like structure. And in this phase or in this S phase or G2 phase, you cannot distinguish individual chromosome because it is in the form of the chromatin fiber, thread-like structure. And once it approaches towards this division phase, this genetic material in the form of this chromatin gets condensed, highly condensed. And in the late prophase, it is having shape like this. And same is the case with this in anaphase. We are having the chromosome as they are separating from each other, as the mitotic spindle attached in this centromere region is pulling these two chromosomes apart. So here we will be having the shape of the chromosome like this. And in early telophase, you can find the chromosome also in this state, but in late telophase, we are having decondensation and chromosome then assumes the shape of this chromatin, just like a thread-like structure. So this structure is only available in this state, this metaphase, and this form, this structure of the chromosome represents the replicated form of the genetic material. That is, this chromosome here is a replicated chromosome, and that is why we are having two sister chromatids. This is after a replication, and this replication occurs during this S phase. And here, for example, if you have chromosome like this in G1 phase, and in late S phase it will be like this, because replication has occurred in this S phase. In S phase, the chromatin material or genetic material gets replicated. But here, this replicated chromosome is in the form of the chromatin fiber. Replicated chromosome in this S phase cannot be distinguished. And it enters into the G2 phase and then finally enters into this prophase and metaphase. During this whole course of this interphase, the chromatin material is just like a thread, and most familiar structure like this is present only in the metaphasic stage of the mitotic division. That is, when you are having two sister chromatids attached at the single centromere, then this chromosome is present at the metaphase of the division. This chromosome is present only in the cells which are going through cell division, that is, which are dividing cells. You cannot find this chromosome, this structure of the chromosome, in non-dividing cells. When the cell enters in the G1 phase, then the cell has decided that it has to go through the division, and this division phase follows this order. Cell enters into the S phase, then into the G2 phase, then into the mitotic division phase. And these stages are highly dynamic so far as the chromatin or genetic material is concerned. And during these different stages, this chromatin or genetic material changes its shapes. And here it is in the form of the chromatin. It is just like a thread-like structure. And in S phase also it is thread-like structure having these nucleosome core particles, and it appears as a thread-like structure. And here also it is like thread-like structure, that is chromatin fiber. And when this chromatin fiber approaches towards the division phase, slowly this chromatin fiber undergoes high level of condensation. And here at this stage, this chromatin is in the highly condensed state, and in the highly condensed state, it appears like this. As it is replicated, so we are having here two sister chromatids. This high degree of condensation, as you know, helps this chromosome to segregate easily in two dividing cells. That is, it is easy for this chromatin, for this genetic material in the condensed state to get segregated, to get separated easily into two cells. If it is decondensed state or in this fiber-like structure, then it will be very difficult for these chromosomes to get segregated. So here we are taking the structure of the chromosome present at the metaphase. You can also take the example of like this chromosome, and this chromosome is a single chromosome, and this chromosome is two chromosomes actually, and these two chromosomes are sharing the same centromere. Here we will describe the structure of the chromosome, and this chromosome is the chromosome present at the metaphase. Another important point so far as the structure of this chromosome is concerned, this chromosome in the structural hierarchy of the genetic material is at the topmost level. And we have nucleotides at the base. For example, we are having these nucleotides. Nucleotides are the basic building block of the genetic information. Like proteins, we have amino acids. That is, amino acids are the basic building blocks of the proteins. So is the case with this genetic information, genetic material. Genetic material is composed of these nucleotides. These nucleotides get interlinked with each other through phosphodiester bond, and they lead to the formation of nucleic acids. So these nucleic acids are chain of nucleotides. Here I am trying to explain you the structural hierarchy of this genetic material. So far as the complexity of this genetic material is concerned, this chromosome stands at the topmost level. And at the most simplest or basic level, we are having these nucleotides. And these nucleotides, when interlinked through phosphodiester bond, they form a chain of nucleotides, and that chain of nucleotide is called as the nucleic acid. And here we are having two nucleic acids. One is the RNA and second is the DNA. Here this nucleotide acts as a building block, but so far as the individual identity of this nucleotide is concerned, it is meaningless. That is, this nucleotide at individual level is not having any meaning. For example, if you consider the deoxyadenosine monophosphate, this nucleotide is not having any meaning. Similar case is with this uracil or guanosine monophosphate. So these nucleotides at individual level are meaningless. But when you combine these nucleotides in this order AUG, these are having some meaning. That is, in this combination, these nucleotides, three nucleotides, will encode the methionine. That is, at individual level, these nucleotides are meaningless. When these nucleotides are combined in a specific sequence, and they are combined in a specific sequence in nucleic acids, and nucleic acids can either be the RNA or it can be the DNA. And for example, we take example of this DNA. In this DNA, we are having these nucleotides arranged in a specific sequence, same like amino acids in the proteins. And as you remember, in the proteins, at the primary level or at the primary structural level, we study the sequence of amino acids. That is, if you consider the sequence of nucleotides in the DNA, then you are actually defining the gene. That is, for gene, most essential part, most primary part is the sequence of these nucleotides. That is, at primary structural level, we are concerned with the sequence of nucleotides in this nucleic acid chain. And here, this sequence of nucleic acids at the primary structural level is defining the gene. So here, if the nucleotides are present in a specific sequence, that is gene. Or you can say this naked DNA without proteins, without any supporting proteins, is gene. That is, gene is the naked DNA. Proteins are essential so far as the expression of gene is concerned. Proteins are involved in the expression of the gene. But primarily, gene is the sequence of nucleotides, and this sequence of nucleotides defined as the primary structure of this nucleic acid. And as you know, this DNA forms double helical structure. Double helical structure I described at the Watson and Crick. That forms the secondary structure of this DNA. Now we have primary structure and secondary structure of this nucleic acid. Or for example, in case of this DNA, we are having the primary structure. This primary structure defined as the gene. We are having the double helical structure of this DNA, that's defined as the secondary structure of this DNA. DNA present in eukaryotic cells, if extended out or if stretched out, is larger in size as compared to the size of the cell where it is supposed to get stored. For example, in case of bacteria, if you extend or if you stretch the genetic material, its size is very large as compared to the size of the cell. Similar is the case with the eukaryotic cell. In case of humans, we have, if you extend the genetic material present in a single cell, its length is almost 2 meters. So this 2 meter length is very large as compared to the size of the cell. So what we require this DNA or this genetic material should be packed tightly so that it can fit into the smaller size cell or nucleus. So we use here this DNA or this genetic material or this nucleic acids. They are being associated with the proteins, and once this DNA or genetic material gets associated with the proteins, and these proteins, as you know, are being used as a packaging material. And most common proteins which are being used are the histone proteins, and this association with the protein is also essential for the regulation of this gene expression. So these proteins can either be used for the packaging or it can be used for the regulation of the proteins. So in short, we are having this this naked DNA gets associated with the proteins. When this naked DNA gets associated with the proteins, we are having this structure that is called as the chromatin. Now this chromatin is DNA plus proteins, and this chromatin is present, as I described it earlier, in the interphase. That is, when a cell is going to divide, this genetic material remains in the form of the chromatin. And this chromatin, as you know, we are having the nucleosome core particles arranged like in linear fashion, and DNA is wrapped around these nucleosome core particles like this. Chromatin fiber appears as the beads on the string. This is the basic structure of this chromatin. And here we are having secondary structure up to the double helical structure of this DNA. And remember, this is the naked DNA without having any protein associated with it. Tertiary structure of the genetic material is when this DNA gets associated with the protein to form this chromatin, and higher level of folding forms the tertiary structure. That is, the tertiary structure starts from this chromatin structure when this DNA forms higher level of folding along with this proteins. So at the basic level, we are having this chromatin fiber. This fiber having these nucleosome core particles arranged in linear fashion, and this DNA is wrapped around these nucleosome core particles. These nucleosome core particles are composed of, as you know, histone proteins. So these histone proteins form the nucleosome core particles, and then DNA is wrapped around these nucleosome core particles, and this forms the chromatin fiber. And at the base level, we are having 10 nanometer fiber of this chromatin. This is the basic level of this chromatin. Then this 10 nanometer fiber forms the solenoid of the 30 nanometer fiber. Then it undergoes higher level of this folding. Finally, this chromatin leads to the formation of the chromosome. So this chromosome is actually the highest condensed form of this chromatin. That is, when this chromatin undergoes higher level of condensation, then it leads to the formation of the chromosome. Now you can understand how the structural hierarchy is arranged. At the base level, we are having this nucleotide, and as I said, this nucleotide in itself at an individual level is meaningless. It is not having any meaning. When this nucleotide is arranged in a specific sequence, then only it can have a meaning. Nucleotides are arranged in a specific sequence in the nucleic acids, and we are having two nucleic acid, RNA or DNA. This DNA, when we consider the sequence of these nucleotides, then it defines the primary structure of the DNA. And this primary structure refers to the definition of the gene. And at the secondary level, we are having this double helical structure of the DNA. This DNA, as you know, is very large in the size. So this DNA has to be packed very tightly so that it can get fit in a very smaller space like nucleus or the cell. So this packaging is done with the help of these proteins. When the DNA gets associated with the proteins, it leads to the formation of the chromatin. And at this level, we are having the tertiary structure of this genetic material. That is, this tertiary structure refers to the higher folding of the DNA in this chromatin. So this chromatin, when it is folded or when it is condensed highly, then it forms this chromosome. Usually if you consider the cell cycle, we are having here the division phase, then we are having the G1 phase, and here it is the S phase, and this is G2 phase. Now if you take these structural hierarchies, nucleotides, nucleic acids, chromatin and chromosome, remember that the genetic material always remains in this form or in this form. That is, genetic material is either in the form of the chromatin fiber, and in this chromatin fiber we are having either 30 nanometer fiber or 10 nanometer fiber, or we are having this 30 nanometer fiber. So this genetic material during this whole cell cycle is either in the form of the chromatin or in the form of the chromosome. So this chromatin, you can say, is the extended form of the genetic material, and this chromosome is the highly condensed form of the genetic material, and this chromatin is present during this interphase, G1 phase, S phase and G2 phase. In these phases, the genetic material is present in the form of the chromatin. When the cell enters into the division phase and in the metaphase, this appears as the chromosome. In the chromosome form, this genetic material is in the highly condensed state, and it is in this state that the chromosome appears like this. And we are having this chromosome having these structures. One is the telomere, then we are having the centromere, and this chromosome structure here refers to the structure of the chromosome present in the metaphase, that is metaphase of the mitotic division phase. So let us try to understand what these structural elements are meant for, and what are their functions and what are their structure.